Good afternoon, everyone. Thank you for having me today. I'm the first year PhD student in the Institute for Neurosciences at Montpellier in Cedric Rolf's team. And today I'm going to present my work on contribution of neutrophils to amyotrophic lateral sclerosis pathogenesis. So recently, human dysregulation have been increasingly suspected to play a role in ALS, with neuroinflammation being strongly implicated, particularly through the activation of microglia and lymphocytes.
But neuroinflammation is also characterized by the infiltration of immune cells, including T cells, monocytes, and macrophages, which can play a protective role in ALS. However, little is known about neutrophils. Several studies have reported a higher proportion of neutrophils in circulating blood of patients, along with an increased neutrophil to lymphocyte ratio. These results have been linked with a faster disease progression and a shorter survival. Neutrophils are often overlooked cells of the immune system, but they are the most abundant leukocyte in the human blood. And when damage occurs, they are the first line of defense and the initiator of the inflammation. Neutrophils used different mechanisms of actions like degranulation, phagocytosis, and netosis, which is a mechanism where the DNA and antimicrobial molecules are released in the environmental extracellular environment, forming what we call neutrophil extracellular traps or nets. Under normal conditions, inflammation is controlled and contributes to the protection of the body. But however, when the immune system become dysregulated, neutrophil function and particularly netosis have a deleterious effect on surrounding cells. So netosis have been shown to play a role in several diseases, including neurodegenerative disease, where netosis contributes to neuronal damage. So based on all these results, we wonder whether neutrophils could also participate in neuroinflammatory processes observed in ALS. So previously in the team, we analyzed the expression of different markers of neutrophils activation in the circulating blood of patients. And the team identified, population expressing CD16 and CD10 like mature, normal mature neutrophils. But this population do not express the CD62L marker, and CD62L negative neutrophils have been identified to be immunosuppressive population capable of inhibiting T cell response, suggesting that neutrophils might contribute to immune regulation during ALS-related inflammation. So to explore this further, we used the SOD1G93A transgenic mouse model. These mice develop ALS-like phenotype with an onset occurring between day 18 to 100 and characterized by the beginning of motor neuron degeneration. Using this model, we performed flow cytometry, analysis on blood, spinal cord, and sciatic nerve of wild-type and SOD1 mice at different time points corresponding to the disease development. We use a panel of several antibodies that allow us to identify neutrophils and follow them along with the progression of the disease. So in blood, after analyzing neutrophil within the CD11b positive myeloid cells compartment, we observe a trend toward an increased frequency of neutrophils in SOD1 mice, mainly at the early stage of the disease. And in the sciatic nerve, at day 40, we identified a subpopulation of neutrophils expressing immunosuppressive character like MHC2 marker that could enable them to inhibit T cells response, similar to the population previously identified in patient blood. Next, we analyzed the immune infiltration in the spinal cord. We analyzed the neutrophil relative to immune and microglia cells, and we could see an increase of frequency of neutrophils in presymptomatic stages, following by a decrease of this frequency that corresponds to the augmentation of microglia during the pathogenesis. This suggesting a shift in the immune cell composition as the disease progresses. Following all these results, we investigate the susceptibility of neutrophils to undergo netosis upon activation with PMA. So we isolated neutrophils from the bone marrow of wild-type and SOD1 mice. We cultured them and stimulated them with PMA. And after we fixed them and immunostain the cells with anti-myoperoxidase and anti-citrulline H3 antibodies to mark the netos. And after quantification of the netosis, we could show that neutrophils coming from SOD1 mice are more prone to form nets compared to the wild type. To assess the cytotoxicity of the neutrophils, we performed co-culture of motor neurons and stimulated or not neutrophils with PMA. So we isolated neutrophils from HB9 GFP mice and cultured the motor neurons for during several days, and then added the neutrophils stimulated or not, and we followed the motor neurons living the survival using live imaging. So so the motor neuron alone or with neutrophil alone or with PMA alone, we show no differences in the surviving and the percentage surviving motor neurons. But when we added neutrophils with activated with PMA, we show a marked decrease of the surviving motor neurons, showing us that neutrophil alone has not an impact on the surviving motor neurons, but the netosis from the neutrophil PMA activated have a deleterious effect on motor neurons survival. Following this observation, we investigate the presence of nets in the spinal cord of mice. So we performed immunostaining using LY6G antibodies for the neutrophils and anti-MPO and anti-citrulline H3, sorry. And we show colocalization of these three marked cells and confirming the presence of nets in the spinal cord. So all these results showed us an increased frequency of immunosuppressive neutrophil in ALS patients, a trend toward an increased frequency of classical neutrophils in the blood of mice, and an immunosuppressive neutrophil subpopulation in sciatic nerve. We also show a neutrophil number increase in presymptomatic stages in SOD1 mice. And about the cytotoxicity, we could show that ALS neutrophils show increased susceptibility of netosis, that only activated neutrophils trigger death of motor neurons, and that there is an evidence of neutrophil extracellular DNA traps in SOD1 mice. All these results allow us to conclude that neutrophils in the ALS pathogenesis have a dual role with anti-inflammatory and pro-inflammatory properties. Thank you, everyone, for listening to me. Of course, thank you for my team and our collaborators, and thank you, RLSLA, sorry, for the funding of this project.
Thank you very much. Are there questions? Then I have a question. Is there evidence on migration or immigration of neutrophils in human ALS tissue? Has this been checked already?
We didn't check on human tissue yet, but it's in the project. But it's not easy to have human tissue for this type of staining. But yes, it's a question we have also.
We have one question.
It's nice work, nice talk. It's a naive question. Do neutrophils interact with macroglia also?
It's one of our hypotheses, yes, because neutrophil can release like the cytotoxic chemicals in the environment, so it could activate macroglia at some stages of the disease. It has to be checked, of course, because of this tendency of activation of macroglia during the disease.